Okay, so good morning to everybody. I switch to English. And we continue with our course, Pharmacology and Physiology of Neuropsychiatric Disorders. And today's meeting will be designated to the topic interaction between classical monoamines and trace amines. I think I will say, to my opinion, it should be one of the most interesting topics we go to cover in this course. I myself totally amazed about trace amines. I was started to work with them. I get aware about the existence not only a few years ago. I think we already spoke about trace amines. They're actually monoamines. It's a monoamine molecules. Why? Because they trace monoamines, because they present in the brain in trace concentration. The concentration of trace amines is at least 1,000 times less than the concentration of the classical monoamines, such as dopamine or adrenaline or serotonin. How they come to existence? They synthesize as side products during the synthesis of classical monoamines. For example, tryptophan. Serotonin is synthesized from tryptophan, but also same tryptophan first turned to tryptophan by tryptophan deoxygylase to 5-hydroxytryptophan. Then it's later go to serotonin. But if tryptophan straight away taken by aromatic amino acid decarboxylase, it's turned to tryptamine, which tryptamine is one of these amines. And each classical monoamine, so-called classical, what does it mean classical? It's not music, but classical is because we knew about them in advance, much before that we first detected and isolated trace amines. Classical monoamine has a trace partner. Serotonin, tryptamine is a trace partner of serotonin. Dopamine have partners pia, tyramine, and octopamine. Actually, dopamine and noradrenaline, all catecholamines, they have this catecholaminergic trace amines. Because their concentration is so low for they've been known, I think, for something like 50 years, even more. Trace amines, they were first isolated from the brain and peripheral tissues. But before their concentration is very low, they were believed to be the side products of the synthesis of classical monoamines. And as a side products, they were believed to be, and because their concentration is so low, it was considered that unlikely that they have biological function. But what's happened later, in late 20 years, receptors to trace amines were detected, isolated from the brains. It was really very amazing discovery, because molecule which has so little, so small concentration has different types of receptors. There are 9 genes for the trace amine associated receptor, but not all of them, I think 5 or 6 out of them end up in protein. Out of trace amine associated receptors, the true interest was in trace amine associated receptor 1 and why? Because it's densely expressed in the limbic area of the brain. You can find in the prefrontal cortex, in the amygdala, in the hippocampus. And what is extremely interesting, you find in ventral segmental area and in dorsal raphe nucleus. You remember what are these brain areas? Dorsal raphe nucleus and ventral segmental area. Exactly. It means that it's already make some hint that trace amines, they might interact with classical monoamines. I had first to not to show it. Last question. What do you think if the concentration of trace amines so low, how can they be detected by the receptor? Yes, affinity, but also where do you think this receptor is located? It was very amazing also. Actually, the TAR1 receptor can be located on the cell membrane, like all other receptors. But was very interesting that this receptor most of the TAR receptors, trace amine associated receptors, they located within the presynaptic neuron. It means it's not a classical neurotransmitter which released from one neuron and affect from another neuron. Probably most of the transaminase trace amine associated signaling happens within the same cell. This is released from it's being synthesized in presynaptic neuron, and probably its main area of action, main targets also associated within the presynaptic space. Why we need such a mechanism which act on a presynaptic level, we will hopefully see later. Later it was, yes. Sorry. Top in the Zoom. I have Zoom. I'm recording.
You might have the waiting room or something.
No, I did not announce. Somebody want to join in Zoom? Well, I did not know. How can I can send you link, but how can I do it? Maybe we'll just see the recording later. Sorry for this. Next time I will make a link to before presentation. What is more amazing, in recent years it was discovered that trace amine associated receptors, they have I start to lose pointer in the middle of the presentation. I really don't know why. Yes, I have it back. They have antidepressant ligands, artificial exogenous agonist of trace amine associated receptor 1. They have antidepressant-like effect, antipsychotics-like effect, and what is very interesting, it has also anti-addictive effect, which we did not see with then one molecule combined antipsychotic and an antidepressant-like effect we saw before, atypical antipsychotics, but we never saw before molecules which also combine anti-addictive effects. Let's start to speak about antidepressant-like effect of TAR1 ligands. These are two molecules which were prepared within Hoffmann-La Roche. One, I will call them by short name, Ro90 and Ro97. One is a full agonist of trace amine associated receptor 1, and one is a partial agonist. And this is a classical behavior test, which we already met several times in our course. It's a forced swim test in rats. And we saw that a full agonist does not have effect in swim test on immobility time, but partial agonist Ro97 has a pretty strong antidepressant-like effect, emphasized by the fact that it decreases the immobility time during the swim test. It's very interesting why full agonist, which is supposed to be more potent molecule, does not have effect, and why partial agonist does have effect. In humans, both have, I think, antidepressant-like effect, but after chronic administration. Another partial antagonist, partial agonist to Rho48, and it also has antidepressant-like effect in rat, emphasized by immobility time during the forced swim test, but it has also positive effect in another. It's another test for antidepressant and anxiety-like behavior in rodents. It's stress-induced hyperthermia. Acute stress, it's typically induce body temperature because of the cytokines and immune reaction. But if animal is less anxious, the stress-induced hyperthermia will be also less. Now I go to speak about something interesting about depression and sleep. I have reminded in the beginning that one of the symptoms of depression is a sleep disturbances. It can be either hyposomnia or hypersomnia. But more common in depressed patients, they actually sleep, but they complain, but they feel that they don't sleep, or they feel they don't get enough sufficient rest from the sleep. And why it's happening because of change of the sleep architecture. You know all about probably sleep stages, sleep architecture, which stages we have in sleep. Yeah, technically, the most important is REM and non-REM. Non-REM also have different stages, but for us, for context of depression, is more common of REM. In depressed patients, REM sleep, onset of REM is earlier. We need healthy patients, we need to sleep a couple of hours to start to experience the first REMs. And first REMs are very short. And towards the end of the dream, after many hours, close to morning, we start to experience this long REM periods. And depressed patients, the onset of REM is very fast, and they see immediately they experience immediately pretty long episodes of REM. And what's happening if we treat animals with R1 ligands? This animal will treat it again with a partial agonist with Rho97. We have more time awake, these animals. They have much less REM sleep, and they have increased REM latency. REM started to be, they start to experience REM later, and they have also decreased REM to non-REM sleep ratio. It means that amine-associated ligands, they may have positive effects, they may repair abnormal sleep architecture experienced by depressed patients. Let's speak quickly about antipsychotic-like effect of trace amines-associated ligands. I go to show you something a little bit different type of data. There are also behavioral data on behavioral model for psychosis and for antipsychotic-like effect. But to be honest, it's pretty questionable because psychosis, which is a purely cognitive disorder of thinking, and since we don't understand animal thinking, it's very hard to model psychosis in animals. Most of the models of psychosis in animals, it's psychotic-like behavior, hyperactivity induced by stimulants like amphetamine, but I put this to most of the section of anti-addictive effects. But what we can do, how we can compare novel antipsychotics with under development like this R ligands with a classical, we can perform functional MRI, and it's more specific, a special type of functional MRI. It's called pharmaceutic fMRI. And what this researcher in this paper did detected that trace amine associated agonists, they have similar effect on they activate similar areas of the brain like atypical antipsychotic olanzapine. The areas like nucleus accumbens, dorsal striatum, piriform cortex, medial prefrontal cortex, and etc. Who are interested, I uploaded this paper to the course website, all papers about trace amines. I very recommend you. I unfortunately in my career I did not manage to spend time with a brain imaging, but I strongly recommend to you if you have opportunity to learn it because it's really something that allow us translate results from basic animal research to the clinics. And let's see about anti-addictive effects, which is more interesting because, to my opinion, the first molecule with a first potential drug with a clear anti-addictive-like effects. Here, most of the anti-addictive-like effects of TAR1 ligands, they against stimulants, not I never heard about alcohol or about depressants, but stimulants like amphetamine, cocaine, they pretty strong antagonistic effect. Here is the result of the test. It's called locomotion. Somebody of you familiar with this test? Yeah, nice. It's actually a very simple test. It's very simple but very informative test. Technically speaking, you just put rat or mice to the box, to the open field, and you see how animal is behaving, how much is moving. You can also check parameters like how much red spent in the center and in the periphery. What you think? What it will be informed? What it will indicate? Center and periphery. Exactly. And more anxious red spend where?
Around the walls.
Yes, naturally. Like we are also the same. And also can take parameters like climbing to the wall. But in this case, they simply checked locomotion and animals. They have groups of animals. One is a wild type animals, WT, and one transgenic animals, which was overexpressing TAR1 receptor. So it's not pharmaceutical treatment with the TAR agonist. It's just animal with has a lot of TAR1 receptor, more than normal animal. And animals that were injected either amphetamine or vehicle. If animal were injected vehicle, no change in locomotion happened. If wild type animal were injected amphetamine, you see huge increase in locomotion activity because it's a stimulant that induces hyperactive behavior, and it's a typical response, like in all species, to amphetamine. But what happened when you injected amphetamine to transgenic animals overexpressing TAR1 receptor? They have much less, they also have had significant increase in locomotion, but it was significantly less than in wild type animal. It means that TAR1 receptor, activation of TAR1 receptor blocks an opposite effect than amphetamines. Yes. Yes. Thank you for good notice. It also not only, I think they did not compare it. They make final comparison only by to distance, but not time of the effect. But yeah, very good point.
Why is the 75 mark dollar sign of
Excuse me?
the 75 mark of the transgenic amine is it's a dollar sign, but it's
We need to go to paper. I believe that the star is a significant difference from baseline, and dollar is significant difference between genotypes. I think so. We need to go to paper to check it. This is results with this is another partial agonist. Actually, we already Rho 48. Actually, we already met with this molecule in previous slides. And Rho has also antagonistic effect on cocaine-induced hyperactivity in rat and in mice. If you pre-treat rat with this ligand, and then you apply cocaine, if you apply cocaine and then you treat also animal with this ligand, it antagonizes the effect of cocaine on the locomotion activity. But what is more interesting, it's also TAR1 ligand. If you can say it's if it's decreased locomotion activity after amphetamines or after cocaine in rodents, it doesn't make that it will help against addiction, because people they take stimulants not to run, they take it to experience reward. And how we can check it in animals? Some of you know self-administration protocols. Well, I see behavior. I think most of you know better than me. We can we can learn rats to inject themselves cocaine. Rats, they have catheter implanted usually to jugular vein, and they placed in the cage with two pedals, one active, one inactive pedal. Rat is a very curious animal, start to touch and to learn everything in the cage, and soon almost every rat learns that if you touch this, touch this pedal, then you get high, then you get dose of cocaine, and they press more and more to the active pedal. But you see, if you treat animals with Rho 48, they really lose their preference and lose their motivation to press the active pedal. It means that they lost the hedonic feeling after cocaine, and they lost also their wish to re-experience. Question which we asked in the beginning: how do you think TAR1 receptor works? If its mechanism totally different from classical monoamines, or its mechanism which still involved classical monoamines? What do you think? Should we vote? Who thinks that it's involved trace amines? And who thinks that it's totally different mechanism? Well, most of people abstained. Okay. Answer to this question, if it's involved trace amines or if it was classical monoamines or not, we will go to, first I will suggest that it does involve classical monoamines. There are three reasons why we suspect it. First reason, that endogenous trace amines, endogenous ligands of TAR1 receptors, they synthesize in monoaminergic neurons. Right? Yes, because they metabolically connected because they connected to classical monoamines. I don't think that they can be synthesized in other type of neurons. It's the first reason that there might be some interaction. Second reason, we learn that traces the TAR1 receptor, it's an intracellular receptor, and it's acting on presynaptic level. It means if trace amines synthesizing monoaminergic neurons, they should also work on monoaminergic neurons, mostly. And indeed, we know they are densely expressed in monoaminergic nuclei. There are three reasons which suggest us that it should be strong functional interaction. But we need to check it experimentally. And it was experiment. We cannot conclude something only because it's looking very logical and very obvious. It's always need to be experimentally. It was indeed first checked by in vivo and ex vivo electrophysiology. And later on, with involvement in my laboratory, we also checked it on level of in vivo electrophysiology. Let's see first ex vivo or in vitro electrophysiology results. This is interesting experiment from the Hoffman-LaRoche. They took Ro17, it's another full agonist of TAR1 receptors, and they applied the brain slices. Animal euthanized, brain removed, you cut the brain to the thin slices. Within the slice, you recognize brain areas that you wish to work in, like dorsal ventral tegmental area, dorsal raphe nucleus, and locus ceruleus. And what they found. This is control conditions, firing of dopamine neurons in wild-type animals, transgenic animals. Firing was much faster than in control animals. It means that TAR1 tonically stimulate activity of dopamine neurons. Yes, it's overexpression, but let's see something very paradoxal. When you apply full agonist, it should even more excite them, but no, you see decrease. And when you apply an antagonist, this is how it's called, EPPTB. It's an antagonist of TAR1 receptor. It's reverse the effect of agonist. Same with dorsal raphe nucleus. Serotonergic neurons in transgenic animal overexpressing TAR1 receptor. They're more active, but when you put agonist, activity goes down. When you put antagonist, activity is recovered. And similar picture you looked in the locus ceruleus, in the noradrenergic area. It's another study with a partial agonist and control animals. And here is something very interesting. It's a partial agonist. Partial agonist, more or less, it works like agonist in normal condition, but it's in comparison with the full agonist, it's becoming like antagonist. Here you see control animal. They applied partial agonist, and they get increase in the firing activity. After washout, what you can do in vitro, but you can do in vivo, you cannot wash out the compound easily. The activity comes back to the basal level. Same in the dorsal raphe nucleus. This is a control. Increase after partial agonist and coming back to basal firing activity after washout. And these are activity in another transgenic animal. First, we saw transgenic animal which was overexpressing TAR1 receptor, and this is knockout. And because there is no knockout receptor, and there is no effect of partial agonist.
Is it most important expression in with the base firing?
I'm not sure. It seems. I think not. I think knockout. They don't have increased firing activity, but you cannot. I think they have different time scales here and here. I don't think that you can combine them by this figure. I was also thinking about it, but looks like not. And finally, what they check in this paper, it's more interesting will be in with respect to our next class. Class will be next meeting, but maybe we even started today. They checked the effect of TAR1 on GABA neurons. GABA neurons, which are also located in dorsal raphe and ventral tegmental area. And what they found that in wild type animals, RO90, RO17 full agonist did not have effect, but it did have a suppressing effect on the GABAergic neuronal firing activity in transgenic animals, which was reversed by antagonist. It says that what we said before, that trace amine receptors, they work only on presynaptic level, is not totally true. They also expressed and also somehow worked on some type of postsynaptic neurons like GABA neurons. Here we came to vivo experiment, which we performed in my lab. It was very nice collaboration between Hoffmann-La-Roche in the Hoffmann-La-Roche Research and Development Center in Basel, Switzerland, and our Center of Bioscience, which you were our guest a week ago. And this collaboration started. I attended a meeting of American College of Neuroscience Pharmacology in 2018. It was a very nice place in Hollywood, but not Hollywood, California. There is also Hollywood in Florida, less known, but extremely beautiful place. It's very hard to work and to listen to lectures when you come to such a fantastic place, but I tried my best to do it. And I met this guy. I get Marius Höner. He was head of the lab, synaptic plasticity lab. Now he is a vice president for neuroscience research in Hoffmann-La-Roche. And we discussed about collaboration. I saw his presentation. He presented these results on trace amine associated receptor 1 and firing activity in vitro in slices. And I told him, do you wish us to check it in vivo? Because in vivo, sometimes you are very surprised when you check the same neurons, but not in slice, but in the full intact organisms, and there's very different results. He said, okay, that was very fruitful collaboration. And what was our in vivo results? In vivo results, when we apply, we also checked RO90 and RO97. You remember, RO90 is a full agonist, and RO97 is a partial agonist. And hydroxydipat is antagonist of 5-HT1A receptors. These are serotonergic neurons. When we apply RO90, the full agonist, we have decrease in the firing activity of serotonergic neurons. It's more or less same like Marius get in transgenic animals in vitro. But when you apply after RO97, it's also dose-dependently recover the inhibition induced by RO90, which makes sense because RO97 acts as in the presence of antagonist, in the presence of full agonist, it acts as antagonist. Same picture was with dopaminergic neurons. We get decrease of the firing activity of dopaminergic neurons after 90, and we have recovery after Rho97. This also makes sense. With noradrenaline, we were surprised. We still don't know fully to explain it. In vitro, it was effect of noradrenaline. In vivo condition, we checked and double checked it, and we did not get statistically significant effect of TAR1 on ligands on noradrenergic receptors. But the main surprise waited us when we tried chronic administration. Before we saw acute administration, but here we treated animals with Rho90 chronically for 14 days. And after 14 days, we checked serotonergic neurons. We did not see difference in the firing rate, but we saw increase in the burst activity of serotonergic neurons, which was. Then we checked dopaminergic neurons, and we were surprised even more, because in dopaminergic neurons, we get after chronic, we get opposite effect from acute. We get increase in the firing rate of dopaminergic neurons and increase in their burst activity. And how would you explain the difference between chronic and acute effects? Any ideas? Exactly. Yes. Well, we still don't know it, but it looks like Rho90 and Rho90, after you administer it animal, it's increased extracellular serotonin. It was found in another study. I had to bring the study, but unfortunately I did not. And because of increase in extracellular serotonin, it's activate 5-HT autoreceptors, which expressed a 5-HT autoreceptor on serotonergic neurons and 5-HT2A receptors on GABA neurons, which innervate dopaminergic neurons. And inhibitory effect mediated via 5-HT1A receptor and 5-HT2A receptor is more strong than excitatory effect of TAR1 receptor. It's why we have decrease in the firing activity after acute administration. After chronic administration, both receptors desensitize, and it's very interesting because with our previous treatments, we did not see desensitization of 5-HT2A receptors, but with chronic treatment with trace amine associated ligands, probably they do desensitize. In case of serotonin, we see no change in the firing rate, but have increase in the burst activity. In this case, with dopamine neurons, we have increase in the burst activity and firing activity. This is very nice hypothesis, but when we submitted this paper and we were asked to prove this hypothesis, there are two ways, and we did very simple. We didn't have that much time, and we did very simple experiment. You remember this PCP from one of the previous classes? You remember what is this PCP? It's an inhibitor of 5-HT synthesis. If you pre-treat animal with PCP, they get 80-90% decrease in their serotonin content. And what's happened? We make this is the repetition of the previous graph. When Rho90 dose dependently decrease serotonergic firing activity in the control animals. But with PCPA pretreatment, after PCPA pretreatment, if you inhibit serotonin synthesis and the animal do not have any more serotonin, that Rho90 does not have any more inhibitory effect on the firing activity of serotonergic neurons. It's partially support our hypothesis, but to fully support it, we indeed need to do experiment. We need to first treat animal with a TAR1 ligand, and then to administer agonist to check response to agonist and antagonist to 5-HT1A receptor. We are doing it now. We can conclude that putative mechanism, we can answer partially to our questions, the putative mechanism of TAR1 receptor might involve the enhancement of monoamine transmission via the induction of the burst mode of firing of monoamine neurons. How actually trace amines, trace amino acid, how it can change architecture of the neuronal firing? There are three mechanisms involved in burst firing of dopamine neurons. The first mechanism is involve L-type of voltage-dependent calcium channels and calcium-dependent potassium channels. Second mechanism, how burst firing is mediated, it's also in other studies suggested that it's also L-type of voltage-dependent calcium channels, and specifically subtypes. There are subtypes 1, 2, and 1, 3. And finally, another study suggested the burst firing associated with the G protein mediated potassium channels. We still don't have answer, and we are working on it now. We still don't have answer how trace amines actually modulate burst firing, but we have two possible, but we have some which suggest that it's at least possible. This, for example, the molecular study on the trace amine-associated receptor 1 signaling, but not in the brain. This is pancreatic beta cells. And we see that trace amine-associated receptor 1 indeed interacts with first with the intracellular calcium reservoirs, like endoplasmatic reticulum, and then it modulate NMDA receptor activity. Also, R1 receptor is a cyclic, is a JS alpha S couplet receptor, increased cyclic AMP. And another study from different cells, from olfactory neurons, this study not with trace amines, a study with locus ceruleus, but this study suggests us that we can indeed get increase in cyclic AMP can indeed modulate L-type calcium channels. It means that technically speaking, because of the signal transduction mechanism, trace amine-associated receptor can induce burst firing. This induce of burst firing is more likely calcium-mediated, but we still need to confirm and to work on this mechanism, and I do it in collaboration with the Gabriakov lab, which you also visited a week ago. Finally, this is our working hypothesis, and we are working on how we think that trace amines they work, why we need them in the brain despite the very small concentration. On the left, you see serotonergic neuron, schematic schema of serotonergic synapse. On the right, you see schematic illustration of neuron. As we said, most of the tryptophan normally form to serotonin. Serotonin aggregated in the synaptic vessels, and when action potential arrives to the nerve terminal, it's released to the synaptic space. What happened? But some of the serotonin turn to tryptamine. Tryptamine tryptamine affect intercellular place amine-associated receptor 1 receptors, which induce burst firing somehow via mechanism involved calcium. And when firing instead from single spike firing turn to the burst firing, we get increase in dopamine. We get more efficient release of serotonin. It means that this small amount of this trace aminergic partner helps the main monoamine to be released in more efficient ways. This is our hypothesis how it may work. And same picture may exist with PA and octopamine and with dopamine. Okay, we can start a new topic in the time that remained for us, but do you have questions to trace amines? I also want to tell something about this. One of our hypotheses that normal monoaminergic neurotransmission depends on the proper balance between trace amines and classical monoamines. Indeed, concentration of trace amines very low, is 1,000 less than it's up to its attomolar concentration, 1,000 less than the femtomolar concentration of the classical monoamines, but we still need them for proper function of the monoaminergic neurotransmission. When this balance changed from some reason, and we probably start to have less efficient serotonergic and dopaminergic neurotransmission, and then it might be linked with the symptoms of depression. And what, to your opinion, what should be behind this balance? The network of enzymes. If we change activity of one of these enzymes, we can change the balance. And actually, not all enzymes. Here we show only enzymes or only enzyme which synthesize monoamines, but also metabolism very important. Imagine what's happening if you block metabolism of serotonin. You get increase in the serotonin, then you change the activity of this enzyme of aromatic amino acid decarboxylase, which in turn can also change levels of tryptamine. And one of the reasons, maybe one of the reasons why monoamine MAO inhibitors, maybe they is that much efficient because they also can intervene and to correct this balance between classical monoamines and trace amines. It's also one of the up-running topic. Yes, please. Sorry, can you repeat the Yes, anti-additive effect. Well, dopaminergic is more likely because of the. It's a very good question. I will quickly show you on my slide from presentation, which I have 2 weeks ago in Adasa and Karen. Oh, you don't see it? Probably I need to close the original file. Yes, you see it now, but I also need to put it that it will be seen in the recording. Yes, this is a presentation. We will speak about it in the very last classes, but right now I just want to speak about addiction. In addiction, I'm not addiction scientist. Let's say what I say, take it carefully, but generally speaking, there are two main components in addiction. Just a moment. No. How to go to this? Yes, you see it. Actually, two main players in addiction, the dopamine, which is responsible for reinforcement, where you feeling of pleasure and you feel and you willing to re-experience this feeling which you have after the drug intake. And serotonin is behind of impulsivity. But if dopamine is a positively coupled with reinforcement, serotonin is negatively coupled with impulsivity. As much serotonin you have, you less impulsive, you more control your impulses, you're more likely to say to yourself, not despite I have high from it, I don't go to do it. And since trace amines, TAR1 receptor modulate both dopamine and serotonin transmission, it somehow should be connected, it somehow control also mechanisms which are behind of the addiction. Right now it was checked only on stimulants. I don't know if it was not checked, if it was checked like an alcohol, probably not. It's kind of topic for very interesting research. Yes, more questions about trace amines? Okay, let's start. Then we can start the next class, which will continue next time because we still have time. And you want to have break or we will continue to next topic? Okay, next topic. By speaking about trace amines, we probably stop speaking about only monoamines. And now we will start to speak about the interaction between monoamines and other biomolecules. And first part will be interaction between monoamines and amino acid neurotransmitters. We started our course, we spoke first about interaction, autoregulation of serotonergic transmission. Then we spoke about interaction between serotonin with noradrenaline and dopamine. And our talk about interaction between monoamines, other biomolecules, we will start, as I said, with amino acids. There are two amino acid neurotransmitters. It's glutamate and GABA. Both of them, they have same source. Both of them from amino acid glutamine. And what is very interesting in monoamine amino acid neurotransmission, unlike what is make it pretty different from monoamine neurotransmission. In monoamines, we saw that the synthesis happens on the synthesis and metabolism all happens on the level of some neuron, but amino acid secreting neurons, they need astroglia in order to maintain proper metabolism cascade. Nitroglia provide glutamine. Glutamine in both glutamate and GABA neurons, in both of them, they first turn to glutamate. It's irreversible. Glutamate can be irreversibly metabolite within mitochondria, within glutamate neurons. It's getting released and can be also same as the monoamines that can be reabsorbed by transporter which called which called exitative amine acid transporter. And it's aggregated to the synaptic vesicles by different transporter, by vesicular glutamate transporter. But in glutamate, in GABA neurons, glutamate turned immediately, also synthesized, but immediately turned into GABA. GABA aggregated in the vesicle by vesicle GABA transporter, and it can be also metabolized by mitochondria within GABA cells. After release, it can be reabsorbed by GABA transporter back to the GABA neuron, but astroglia can reabsorb both of them. It can reabsorb glutamate and GABA, and it can metabolize them reversibly to glutamate, and glutamate can be reused again to glutamine, and glutamine can be reused for glutamate and GABA synthesis. It means to us that in case that GABA, as I told, that amino acid neurotransmission is very critical. Glia is very critical. Without glia synthesis and metabolism, this transmitter cannot happen. Both of them, glutamate and GABA, both of them, they very old evolutionary, they much older than monoamines. They can found be already in the first multicellular organism in Hydra. It's a Hydra vulgaris. It's very evolutionary ancient organisms. This type of organisms exist for almost 600 million of years, and they already have neural system, so-called diffuse neural system, and this diffuse neural system employ amino acid neurotransmitter, both glutamate and GABA, but most of the neurons in Hydra, they are GABAergic. I personally believe that GABA might be evolutionary first neurotransmitter. Why it can be? Because we don't know right now any organism which has only GABA without glutamate, but technically speaking, theoretically, you can build functional neural system with only GABA, with only inhibitory neurotransmitter. Why? Because of disinhibition, inhibition of inhibition, you get excitation. But it means with a neuronal network, theoretical neuronal network consisting only GABA cells, you still have both types of inhibitory and excitatory signals. But if you will have neural system consisting only of GABA, only of glutamate, the system theoretically will not be able to to produce inhibitory signals. This all before big overview about glutamatergic and GABAergic areas of the brain. Glutamatergic areas of the brain is cerebral cortex, including hippocampus. It's thalamus. It's our more in-brain input for sensory pathways. And this is brainstem. GABAergic of areas of the brain, they are cerebellum and basal ganglia. But what is very important, GABAergic neurons, they located everywhere. Even if the brain area which considered to be glutamatergic, like cortex, we find GABAergic neurons everywhere in the way of interneurons. Also in the dorsal raphe nucleus, locus ceruleus, and ventral segmental area, we found plenty of GABAergic neurons. For example, this immunohistochemistry study from the red raphe nuclei. We found even more GABAergic neurons than serotonergic neurons. And if you do electrophysiology, you more frequently find GABA neurons, which are extremely fast. In the ventral segmental area, you also find plenty of GABAergic neurons. And what is very unique for ventral segmental area, you have the very unique neurons which release GABA and glutamate. One neurons which are releasing both glutamate, excitatory and inhibitory neurotransmitter. Same with locus ceruleus. In locus ceruleus, you have also GABAergic neurons, which are usually found around noradrenergic neurons. And we saw this function, this GABAergic interneurons, they very important in the term of interaction between serotonergic, noradrenergic, and dopaminergic system. We have here serotonergic neurons which project to ventral segmental area and to locus ceruleus, and they activate GABA neurons, which in turn inhibit noradrenergic and dopaminergic neurons. Happen between interaction between H2A and GABA-A receptors for in locus ceruleus and 5-HT2C receptors, and again GABA-A receptors in the ventral segmental area. And we will continue for this next time. I just want to introduce to you. I first get interested in GABA not because of the neurophysiological, neuroanatomic reasons, but from pure clinical and pharmacological reasons. Do you know which drugs acting on GABA you know? Sorry? Yeah, no, it's not really drug. Medicines. Benzodiazepines, exactly. Benzodiazepines, they are very widely very used. They have, for example, clonazepam, which is mostly used for anti-seizures in epileptic patients, but lorazepam and and others, diazepam, they used for patients which are complaining about anxiety, insomnia, irritation, irritability. And many depressed patients, they also get benzodiazepines. And because serotonergic system and GABAergic systems are so interconnected, we really need to put attention how benzodiazepines, how they work in depressed patients. Okay, I think we will continue next time for this. Maybe any more questions? Somebody have question about the final, about the project work and
If you decide to do a presentation, how long should it be?
no, I explain you don't have to. It's not mandatory. Maybe if the course maybe will be next year. Maybe I will think differently in the next year. Right now it's not mandatory, but bonus, and I really recommend it. I don't
How long should it be? Like, people take
5-10 minutes, 10 minutes maximum.